Drive.
Can you put it on Google Drive?
You gotta pause it.
I was gonna say, I don't want to wait for people to get it on Google Drive. I can do this
Is
during.
it going? We have chapter 27, bacteria and archaea. So a few basic definitions. Archaea are single-celled prokaryotic organisms that live in extreme environments such as hot springs and whatnot. Bacteria are single-celled prokaryotic organisms. We all know what bacteria are. So what led to prokaryotic success? Well, the first organisms that ever inhabited Earth were prokaryotes, lived approximately 3.5 billion years ago. And we know that they eventually evolved into eukaryotes through the end-symbiotic theory. Rings a bell. Most are unicellular, although the cells of some species remain attached to each other after cell division. These are some different types of prokaryotes, or bacteria, I should say, either one. Spherical, rod-shaped, and spiral-shaped. So what is on their cell surface structures? You have the cell wall, maintains cell shape, protection, prevents it from bursting. How are these different than cell walls in eukaryotes? Well, eukaryotic cell walls are made up of cellulose or chitin. Bacterial cell walls contain peptidoglycan.
Yeah, you got it.
Which is a polymer composed of modified sugar cross-linked by short polypeptides. Does anyone know which amino acid forms cross-linkages? Carbonyl group? Carboxyl group? Do you know, Sophia? Isn't it the sulfo hydro group or something like that? Okay, so how exactly do we know which shape the prokaryotes take? We use a thing called Gram stain, and that's developed by Hans Christian Gram. They can categorize bacteria based upon their cell wall composition. Samples are stained with a violet dye and iodine, rinsed in alcohol, and then same with the red dye. The structure of the cell wall determines the staining response. There's two different types of gram stains that pop up, gram positive or gram negative. If they're positive, they have simpler walls, large amount of peptidoglycan. If they're negative, they have less of it, and they're more complex. This is an example. If they're positive, you see here they have 30 layers of peptidoglycan, which means they're more complex. If they come up negative, which is like a pinkish dye, they only have one layer of peptidoglycan. And then their cell wall is also made up of these lipopolysaccharides.
Lipopolysaccharides. Jack,
Yeah.
just one thing real quick, go back to your slide. All right, just to kind of add to this, the gram-positive cell wall, because it has several layers of these peptidoglycans, the Gram's stain, which is crystal violet, soaks in very easily. And then when you lock it in place with iodine, it stains very dark blue. Now, the ones on the bottom, which is gram-negative, since they only have one layer of peptidoglycans, but they have that outer lipopolysaccharide layer, it actually resists gram stain, so it doesn't soak in, and that's why we say that it tests negative for gram stain, or call them gram-negative bacteria. The top ones, because they accept the stain so readily, they test positive for gram stain, therefore they're called gram-positive. So once you treat it with alcohol and get rid of the lipopolysaccharides, it will then accept another stain. So they use a counter stain, which is safranin, and that's pink. It's red. And so now you can make a distinction between gram-positive and gram-negative. Does that make sense to everyone? I didn't expect you to know this. I mean, this is stuff I talk about in microbiology.
Any questions? All right, so they have prokaryotes developed a thing called a capsule, which is a sticky layer of polysaccharide protein that surrounds the cell wall. It's a term capsule, but it's dense and well-defined, and allows prokaryotes to adhere to their substrate or to other individuals. It also can protect against dehydration. This is a picture of a capsule around a cell wall. They developed another thing called an endospore, which is used for protection. The original cell will produce a copy of its chromosome and then surrounds that copy with a tough multilayered structure forming an endospore. Most of these endospores can't survive in boiling water. Picture of an endospore.
Now, Jack, do all bacteria produce endospores?
They only need to produce them when they need to develop an extra set of resistant cells in extreme environments.
Right, but are there certain bacteria that can produce them and certain ones that cannot? What's that? You will? Oh, okay.
I don't know.
Wait, so how is this endospore formed? Like, what is it?
Okay, so the original cell makes another copy of itself, and then this copy is then surrounded with another layer, and this layer is kind of put in here as well into the cell itself, which forms another layer of things you have to go through to get into it, kind of just extra
Yeah.
resistance.
What happens is, in adverse conditions, the cells stop dividing, growing, and then it divides underneath the cell wall. The bigger half envelops the smaller half. The bigger half will be called the mother cell. The smaller half will be called the forespore. And then, to cut the story short, once the mother cell secretes these coats around it, secretes peptidoglycans in there, it becomes desiccated, the mother cell disintegrates and leaves behind an unbelievably stable spore structure, which is not a reproductive structure at all. And keep in mind, you have to know that because, like I know on the AP test, if they ask about it, endospores are not about reproduction. It's about survival only in the adverse conditions. So like Jack said, they can survive boiling, they can survive extreme dry conditions. We don't even know how long spores can actually last.
Wait, but is it inside the cell wall? Like,
It's all done underneath
over
the cell wall.
nucleus? It's over, like, the
Right.
nucleus?
So there's a spore, there's coats and a cortex that forms around the spore. And then inside the spore is a copy of the DNA, all the organelles, everything, or not organelles, I should say, all the internal structures, but dried out, desiccated, very stable. And those spores can exist as spores, not doing anything metabolically, biologically inert, doing nothing until conditions are favorable. They found spores in rocks that were very, very old and were able to germinate them by putting them in the right conditions. So we don't know how long spores can actually last, but spores are very, very highly stable. There's two genuses of bacteria that can produce them, the genus Bacillus and the genus Clostridium. Those are the only two that can actually form endospores. No other bacteria can do it.
Moving on, other structures of the bacteria, fimbriae, all right? Some prokaryotes stick to the substrate and one another by means of these hair-like appendages surrounding them. So for motility, about half of all prokaryotes are capable of taxis, which is directing movement towards or away from a stimulus. For example, they move towards oxygen or away from a toxic substance. The most common structure used to move is the flagella. Here's an example of it. You can see it's a tail-like structure. And for further emphasis on motility, we can see that Randy Orton is quite mobile in the wrestling arena.
Okay, so now the role of oxygen in metabolism. We have some different ways that archaea can use oxygen in the way they metabolize. There are obligate aerobes, which they must use oxygen for cellular respiration, and they can't grow without it. there are obligate anaerobes, which they're poisoned by oxygen, and some of these live exclusively on fermentation. There are also there's anaerobic respiration, which is when substances other than oxygen accept electrons at the end of the electron transport chain. And there are also facultative anaerobes, which they use oxygen if it's there, but they can carry out fermentation or anaerobic respiration if it isn't, so it's not really necessary for them. So now nitrogen metabolism. Nitrogen is essential for the production of amino acids and nucleic acids in all organisms. And some cyanobacteria and methanogens convert nitrogen to ammonia using a process called nitrogen fixation. And here, these cells incorporate the nitrogen into amino acids and other organic molecules, and it has a large impact on these organisms. For example, prokaryotes that use nitrogen fixation, they can increase the amount of nitrogen that's available to the plant. And they can't use atmospheric nitrogen, but they can use the nitrogen compounds that these prokaryotes produce from the ammonia. So archaea. Cooperation between prokaryotic cells allows them to use environmental resources they could not use as individual cells. For example, a cyanobacterium known as Anabaena has genes that encode proteins for both photosynthesis and protein fixation, but they can't carry out both at the same time. And for the reason behind this is photosynthesis produces oxygen, which inactivates the enzymes involved in nitrogen fixation. And these organisms here form filamentous chains where most of the cells are found that carry out photosynthesis. But in this chain, there are a few specialized cells that carry out nitrogen fixation, and these are called heterocysts. And each of these heterocysts is surrounded by a thickened cell wall that restricts the entry of oxygen that is produced by neighboring photosynthetic cells. And intercellular connections allow heterocysts to transport fixed nitrogen to neighboring cells and receive carbohydrates. Metabolic cooperation between different prokaryotic species often occurs in surface coating colonies. This is called biofilm. And cells in a biofilm, they secrete signaling molecules that recruit nearby cells that cause these colonies to grow. And they produce polysaccharides and proteins that that have the cells stick to some sort of substrate for one another. And these are common in nature, such for example, dental problems like plaque buildup and such. They can cause a colony, which is actually a colony building on your teeth. So prokaryotic phylogeny. Until the 20th century, systematics-based prokaryotics taxonomy was based on shape, movement, nutritional mode, and gram staining, which is right here an example of the gram staining bacteria. And these are important when rapid identification of a bacteria is needed, but a more accurate method is required when it comes to prokaryotic phylogeny. And applying molecular systematics has produced great results and reveals a large history of these prokaryotes. So versus like with the gram staining in the previous method, we could only tell like what that species, how it, like what was the characteristics of it right now. But with prokaryotic phylogeny, we can look back at its ancestry and see how it evolved over time. And some more diversity. Organisms that obtain energy from light are known as phototrophs. Organisms that obtain energy from chemicals are chemotrophs. There are some organisms that only need carbon dioxide as a carbon source, and these are called autotrophs. And then there are also heterotrophs, which require at least one organic nutrient to make more. So archaea, they share certain traits with bacteria and other traits with eukaryotes, but they also have many unique characteristics. For example, the first prokaryotes lived in extreme environments, and these are known as extremophils. An example of these are extreme halophiles, which live in highly saline environments. So an example of this would be the Dead Sea, and it's the salinity of the water is super high, and if it drops below a certain point, these organisms can't even survive without it. There are also extreme thermophiles which thrive in very hot environments. An example is Archaea in the genus Sulfolobus. And at high temperatures, most cells die because their DNA and proteins denature. But with this organism, they have made changes to their DNA which allows them to survive in places such as these. And these are like hydrothermal vents, which are also found in the deep sea. And an example of this is called strain 121 because they survive in temps up to 121 degrees Celsius. And another example is the thermophile Pyrococcus furiosus, which is used in biotechnology as a source of DNA polymerase and for the PCR technique.
So like what Cameron was just talking about, these type of archaea are methanogens, and they live in areas that have like very little oxygen. That's their name for the way that they can obtain energy. They use the CO2 to oxidize the H2, and then they release methane as a product. That's why they're called methanogens. And they're some of the strictest of the anaerobic archaea because they're really poisoned by the O2. This would be like a typical environment where you find methanogens in like these swampy, marshy areas because they lack the oxygen, so that's why they live there. And these kind of got mixed up. Other places you could find methanogens would be like the guts of cattle or termites or other herbivores because of their diet, and that's why you hear stories about like cows producing a lot of methane gas. It's because of the methanogens living in their guts. They're pretty essential to the breakdown in the animal's gut. They help the digestion. They're also used as decomposers in different sewage treatment facilities. And the ecosystems depend on like the recycling of these chemicals by different living and non-living components in the environment, and all various prokaryotes play key roles in this process. Some prokaryotes would be decomposers, and they break down like the dead corpses, the dead vegetation, and other waste products. That's why the swampy and marshy areas would be like a big hotspot for these types of organisms. Is you think of like a swamp and a marsh, there's a lot of dead, kind of decomposing. That's why you walk in. If you ever been to like a swamp and a marsh, it'd be really hot. Kind of, you just get that feel from it. Now these are all different types of symbiotic relationships. A symbiotic relationship is a relationship between organisms of different species that are in direct contact with each other, and that's called symbiosis. The larger organism would be the host, and then the smaller is the symbiont. So the first type of symbiotic relationship is mutualism, and that's where both organisms would benefit from the relationship. Other relationships would be commensalism, where only one organism would benefit, but the other one isn't really harmed, so it usually doesn't mind that other organism is kind of in direct contact with it. And the last one is parasitism, and that's pretty obvious. It's that one is a parasite, so the other organism is harmed. That's usually you usually think of like a leech latching on, sucking your blood. It uses that kind of parasitic relationship.
And here's a further explanation on the concept of mutualism. You can see the Bautista-Baum combination into an archaea. So they're working together. Okay, here's another example of mutualism is the flashlight fish. So this glowing oval beneath the eye is an organ. Oh, sorry. So the fish uses this light to attract prey. There are actually bioluminescent bacteria there. So they both benefit in that this light helps attract mates and also helps to find prey, and the bacteria receive nutrients from the fish. The best known prokaryotes tend to be bacteria that cause illnesses, but they're also bacteria with positive interactions with humans, and some play essential roles in agriculture and industry. For instance, our intestines are home to about 500 to 1,000 species of bacteria. Many of these species exhibit mutualism, so they digest food that we couldn't do on our own in our intestines. Our intestines can't break them down, so we have these bacteria in our gut that help us. Pathogenic bacteria. So all the pathogenic prokaryotes known to date are bacteria, and they're responsible for about half of human diseases, so these are more along the lines of the bad bacteria. Roughly 2 million people die each year from the lung disease called tuberculosis, and another 2 million die from diarrheal diseases. Some bacterial diseases are transmitted by animals such as fleas or ticks and pests. You can see a tick here. This is Lyme disease. And the most widespread pest-carried disease in this area is Lyme disease. Here's the bacteria of it. It's caused by bacterium carried by ticks that live on field mice. And a rash may develop at the site of the tick's bite, and the rash may be large and ring-shaped, as in this
Yeah,
picture.
the target rash is what they call it.
And Lyme disease, the result of that could be debilitating arthritis, it could be heart disease, nervous disorders, and possibly death if untreated. Here's our review game. In what sense do archaea and bacteria differ? Very general. Tony Barrett.
I would think archaea survive in very harsh conditions.
Exactly, thank you. So they live in extreme environments, bacteria usually don't. And the sticky wall of polysaccharide or protein surrounding a prokaryotic cell wall is called?
It's the one that has 30 layers and a positive gram bacteria, 1 layer and a negative gram.
Alright, it's called a capsule,
Oh, that's wrong.
if it is dense and well defined.